Thank you for tuning in. Our presenter is Jack Regan, CEO and founder of LexaGene. Mr. Regan, you may begin.
Thank you, Jess. So thank you all for tuning in today. As Jess said, my name is Jack Regan. I'm the founder and CEO of LexaGene. LexaGene is a molecular diagnostics company. What we do is we just simplify the process of detecting pathogens. So in this next slide here, this is our disclaimer slide. At a later date. So a quick overview of LexaGene. As I said, we're a molecular diagnostics company. We currently have 35 employees, but we are growing quickly. As of November, we had about $11 million in the treasury, so we still have a very strong treasury. We have commercialized our instrument, which we call the MyQLab. It is a rapid, fully automated pathogen detection system for point-of-need use. So what that really does is it takes the complexity of PCR and puts it at the location where it is most needed, which is really the point of need or the point of care, as we like to say. We have achieved our first commercial sales. So we've gone through the R&D development of the company and successfully manufactured and sold our first systems. A fantastic milestone for the company, but really just the beginning of what LexaGene has in mind for our goals. We have started our FDA EUA study for COVID-19 testing, and we've also started placing systems in the field for our early access program in the veterinary diagnostic space. For those of you who may be curious as to how and why we named the company LexaGene, the etymology of the word LexaGene is provided to the right. Lexa has a Greek root meaning, which means defender of humankind. And gene, we are a genomics company. The genome is made up of many genes, and that's what we detect. So we like to view ourselves as a company looking to protect people, and we do it by really identifying genes and pathogens, and for that matter, any organism you're interested in. We could even do genotyping in the human genome. Next slide here, we're going to go into LexaGene's technology and the markets we are focused on. So when LexaGene was founded 4 years ago, this is obviously pre-COVID-19, at the time, I was very interested in positioning this company to target what we consider to be underserved markets, markets where there's really not a lot of competition. So veterinary diagnostics and food safety are top of that list. Vet diagnostics is often forgotten about. It is a great market. Companion animal diagnostics specifically. People view their cats and their dogs as family members. It's a cash business, a great opportunity for LexaGene, in desperate need of really high-quality testing within vet hospitals. Likewise, food safety, a big demand there. However, coronavirus came along and really, turned the world up on end, if you will, and made us as a company evaluate, okay, what are our plans? How do we best position our shareholders to be happy with their investment in LexaGene, and what's best for the company long term? Human clinical diagnostics has always been on our roadmap. However, we were anticipating entering human clinical after first successfully really establishing a real strong foothold in vet and food safety. But the coronavirus and the FDA's EUA has given this company a great opportunity to get into human clinical diagnostics much sooner than had anticipated, and that's exactly what LexaGene is doing. We're working towards the FDA right now. We're working towards studies to complete our EUA study in human clinical diagnostics, because ultimately the value of this technology is perfectly suited for human clinical. Open access is a fantastic market. I'm going to get into it in a bit in this next slide, but it is considered, in my mind, one of the underappreciated markets out there, and LexaGene really will help define that market moving forward. So this slide puts some numbers on the sizes of these markets. Each and every one of them are sizable markets in their own right. Obviously, human clinical diagnostics has absolutely exploded with COVID-19 testing. It's important to recognize, and we'll get into this later as well, that COVID-19 is not going to just all of a sudden disappear next month. It will be around for years, and arguably it's going to change the way in which diagnostics are performed on a regular basis moving forward. Vet diagnostics is a $4 billion industry by 2023. Again, no competition for in-hospital, animal testing, which is, again, a great opportunity for LexaGene. Food safety, great need for, better detection technologies. Right now, you're likely aware that, food safety problems continue to persist in the industry, and the industry is looking for genetic testing because of its sensitivity, and we believe at LexaGene we can provide that solution. So open access. People don't understand open access. It is not a market that you can go and buy a market research report on. It is really an up-and-coming market, I would say, because right now there are no technologies that specifically target the open access market. And we believe we're going to be the first. And so what exactly is the open access market? It's areas within the industry, life sciences, biotech, pharma, et cetera, that do PCR testing on a regular basis, but they have unique needs. And because their needs are unique, it's very hard for them to get a commercial system to help them. We provide that commercial system because the MyQLab is a fully automated microfluidic device that enables them to automate their own tests. So they have control over what exactly the instrument automates. And we can do it in a very short time frame, effectively removing the human element from testing, which is fantastic. On the right here, we have some of our value props. Our value prop is providing PCR testing at the point of care, and PCR is considered a gold standard. We take that high-quality test, which is typically done in a reference lab, which takes days to return that result, and place that technology in an automated system at the point of care where you can get the answer in just around an hour's time. We also offer multiplexing. What is multiplexing? Multiplexing is the ability to look for many different targets at once. Why is that important? If you look at coronavirus, for instance, you can have multiple viruses causing a fever and a cough. Influenza is one, adenovirus is another, RSV is another. And so during the flu season, you can get confusing results if you get a negative result on a coronavirus test and you don't know if that was negative due to the absence of coronavirus or if it was actually really positive due to something else. And so we can detect many different pathogens at once, giving the clinician, and for that matter, the patient, high confidence that when the result comes back, if it's negative for coronavirus, it should be positive for one of the other pathogens. Our data quality is on par with what is generated at reference labs. We do have a razor blade business model with a high profit margin at volume manufacturing, and we have a strong patent portfolio. So let's talk briefly about human clinical diagnostics, specifically the unmet need in COVID-19 testing. So I'm often asked, what's LexaGene's true value prop? How are we going to compete in this market? It's a very competitive market. We know that. So what are we doing differently? Why would somebody be interested in LexaGene as a product? One of the most salient features for us is, again, going back to what open access offers and the value it presents in combating a disease like coronavirus. If you're following the news, there's been a lot of stories written recently on the UK strain and also the South African strain. Coronavirus is an RNA virus. RNA viruses mutate very, very quickly, and we expect these new strains to emerge. And the important part is we need to be able to contain them. The UK strain is 70% more contagious. The South African strain potentially will evade the vaccines that are under development and are now being administered, which is extremely frightening. We've spent over $10 billion developing and distributing vaccines. If all of a sudden the predominant strain becomes the South African strain, and if it drops the efficacy of those vaccines from the mid-90s down to like 20 or 30%, effectively the pandemic is starting over again. And so here is the important part of our technology where we can contribute. Because our technology is open access, it's very fast for us to put new tests on our system. So we can test for the conventional coronavirus, and we can also look for other strains like the UK strain or the South African strain, likewise flu, RSV, adenovirus, etc. So what needs to happen? NGS, you may have heard of it. It stands for Next Generation Sequencing. This is an ongoing effort to monitor what new variants of coronavirus are out there. When new variants are detected that are a clinical concern, it's very, very important to turn that information in to basically instruments that are generating quality data at the point of care so you have the best possible option to contain the virus before it spins out of control. Right now, our healthcare system lacks this technology, and LexaGene is going to be the only system on the market today that allows for new tests to be very quickly onboarded within days to allow for better containment of novel strains. And this is not going to be something that goes away with coronavirus. More pathogens are going to emerge in the future, whether it's avian influenza or, we just had Zika a couple of years ago. Viruses are constantly threatening, effectively, humanity. And so this is not something which is just going to go away. There will be a continued need for this into the future. So looking at the clinical diagnostics market, please recognize that not all tests are created equal. There are really two big categories of tests. Tests that are done at the point of care and tests that are done in reference labs. The left and the center column here, the isothermal test and the antigen test, these are point of care tests. They're generally easy to manufacture. They're generally fast. However, you can't often have your cake and ice cream too. They have downsides to them. The downsides are generally they have poor sensitivity and they only look for one pathogen at a time. The poor sensitivity is gaining more and more press. If you go onto the FDA's website, the FDA is issuing all of these warnings regarding some of these rapid tests, suggesting that, listen, if the person is symptomatic, you cannot use these tests alone. You have to use them with another test, like a PCR test, to confirm the results. This is because, again, the validity of the test, whether positive or negative, is always in question due to the quality of the test. So recognize that the super fast tests come with a big disclaimer that they don't always give accurate results. The accurate result is generally one that is performed in a reference lab where they do RT-PCR. RT-PCR is considered the gold standard. It's very, very sensitive and specific. It also allows you to quantify how much viral material is there. The FDA has publicly acknowledged the value of doing quantitative PCR. That is the exact chemistry that LexaGene automates on the MyQLab. And the reason that is important is because if you can tell a patient, or if the clinician knows whether or not they have a virus in their system or a little, it provides an indication as to the risk of that person getting sick and potentially, needing, aggressive medical care. It's also an indication as to how transmissible or how contagious this person potentially could be because they have so much virus present. So again, what LexaGene does is it takes that gold standard chemistry, the PCR chemistry that's typically performed in a reference lab, it automates it and places it at the point of care where you remove the time delay in shipping samples to reference lab. And again, reference lab data, you're waiting days to get that information back, which is completely inexcusable in today's day and age. We need to provide fast tests at the point of care so that people know how to conduct themselves and really help contain the spread of this disease. So as I mentioned earlier, this company was founded before coronavirus emerged. Our primary market originally was veterinary diagnostics. It was our primary market because we felt like we could immediately start contributing to this market. There's effectively zero competition for in-hospital testing. The dogs and cats that we all love, they get sick just like humans do, and their pet owners care for them just like their own son or daughter. And so our first test is what we call a bacterial AMR test. What AMR stands for is antimicrobial resistance. So why is that important? So our first offering looks for 20 different targets at once. 7 of those targets are for common bacterial infections. 13 of those targets are for genes that confer resistance to drugs. We've all heard of superbugs. Superbugs are these pathogens that have acquired resistance to the common therapies that we prescribe, and they're becoming more and more difficult to treat, both on the human and the vet side. And yes, these transfer back and forth. So clinicians, whether you are a human clinician or a veterinarian, they want to be able to prescribe the right therapy at the first, sign of when they actually see the patient, and the only way they can do that is by doing upfront diagnostics. And so we provide that, and it will lead to better patient care. It will also allow for better antibiotic stewardship. We have to be smarter about how we're prescribing our drugs. If we continue to prescribe the way we're prescribing right now, the pathogens, these microbes, will continue to evolve and effectively develop resistance. And what was a simple, scrape can turn into effectively a deadly infection. So we really need to be smart about how we prescribe our antibiotics. Doctors, both on the human and vet side, know this, and they are looking for technology to help them understand the antibiotic resistance profiles of the pathogens that they're detecting so they can be better in prescribing their drugs. Food safety, huge, huge need. Right now, food safety is basically dependent on growing up, a culture, which is, very time-consuming, and that time costs food producers money. The biggest need here is in the ready-to-eat food items. These are the ones that have a limited shelf life. Any time spent waiting to get that test result back so they can clear that food product before shipping to the customer, whether it's a restaurant or a grocery store, is effectively lost profit because the less shelf time there is, the less opportunity there is for them to sell that product. So LexaGene is providing a technology to allow them to do genetic-based testing extremely fast so they can clear their products quickly, get them to the customers, and really help mitigate the risk of having a recall. Here we are, open access. This is my baby. I love open access. I think it's a completely undervalued, underappreciated market. And again, it's because there are no technologies until LexaGene. And now I can say this, we've done this now. We've actually launched into this market. We sold our first system into a contract drug manufacturing organization. They came to us asking if we could solve a problem for them. And the answer was, well, what are you looking for? And they were looking to detect a bacteria called C. acnes. And C. acnes is a bacterium that takes about 2 weeks to grow. And this particular bacterium was crashing out their bioreactors, costing them hundreds of thousands, if not millions of dollars of losses per year. And they were looking to PCR because PCR is so sensitive to be able to detect it in a faster time frame. They want to be able to screen their raw reagents going into these bioreactors and test the bioreactors as they're being scaled up from small mini reactors to larger reactors. And these reactors become 20,000 liters in size. And at that scale, it's literally hundreds of thousands, if not millions of dollars of product in those reactors. So this particular company came to us. They came on site. They brought samples. We tested their samples. They were impressed by the data. They looked at everything else out there on the market, and they determined that LexaGene was the best solution for them, and they bought, and we're very excited about this market. We do think this is going to be a big win for us in 2021 and beyond. But it goes much beyond the contract drug manufacturing organizations. There are other markets which really define open access. Anybody who has custom PCR testing needs falls into this category right now for us. That includes pharma, biotech, research, both in academics and in government, and across other sectors like cannabis, agricultural testing. People forget how many billions of dollars a year in crops are lost due to pathogens that affect crops, not humans. And so it's something that definitely, take into consideration. You have botrytis. It causes billions of dollars in losses every year. It's a fungus. It only affects, basically your grapes, your cannabis, and your strawberries. So that's a perfect example of using open access testing to help farmers save, tons of money due to a potential contamination. So next slide here, the predecessor of the technology, a little bit of background about myself. So I am not only the founder of LexaGene, but I'm also the inventor of the technology. My past history is I got a PhD studying influenza biology at UCSF in San Francisco. Went on to work at a national lab. At that national lab, I got involved in developing automated instruments to detect pathogens. Most of the focus was in the biothreat sector. And so we were developing an instrument to detect anthrax, smallpox, and plague, and other select agents. That particular technology was adopted by the Department of Homeland Security and brought under their BioWatch program to do surveillance for a bioterrorist attack. About $20 million was invested in that program, and that system was out there in the field operating. I looked at that system. We built a sister system to that to look at respiratory pathogen detection. That system was fielded up at the UC Davis Medical Center. Those two systems in their own right were great systems. I didn't feel as though they were strong enough to make a great commercial product, so I set out to design a better instrument. It's that new design which LexaGene has now commercialized. It takes some of the principles of these earlier predecessors and adopts those, but it addresses some of their weaknesses, and we've created a very, very powerful instrument which we think is going to have great value in many different markets. Now on to the company financials. So we do have a recurring revenue stream. This is critical for all diagnostic companies. We have two main parts of our consumables. In the upper right-hand corner, you will see a small sample preparation cartridge. That cartridge is very easy to manufacture. It basically is comprised of two pieces of plastic and a couple of filters. Because of its low cost, this is what drives our high profit margin for testing. Every single time you run a sample, you have to burn through one of those cartridges. The operator of the system would load the sample into that little cap next to the thumb of the person loading that cartridge, close that cap, insert it into the instrument, use the touchscreen monitor, and initiate sample processing. That's it. It literally takes less than a minute to operate the system, extremely easy to use, and that really is critical for a point-of-care technology, and it's something that the FDA is very interested in, and they require evaluating that in their studies for point-of-care testing. Down below is our reagent panel. This is a component that goes into the instrument and stays there for a month at a time. You can run at least 25 samples with that reagent pack, and this is also what directs the instrument to look for different pathogens. So you see those 9 different holes at the top of that reagent pack. In each of those holes is a microcentrifuge vial that holds the primers and probes for PCR reactions. Each of those is what we call a 3-plex, looking for 3 different pathogens per microcentrifuge vial. The format of manufacturing those tests is extremely easy, and again, it helps contribute to our high profit margin once we hit volume manufacturing. So, some quick corporate highlights here. Again, we're 35 employees and growing fast. The cap table is again on our website. Take a look at it at your leisure. We have raised a total of $28 million to date, and we're on 2 different exchanges, up in Canada and the TSX, and down the States on the OTC. These data are, again, available on our website, so feel free to look at them. If you have any questions, I'll get you in touch with our CFO. Next slide here, team. Company is, half technology, half team. Who's bringing it to market? What's their experience? Have they done it before? Can they do it again? I'm very proud of the team we've assembled. Top row here, we have executive directors and officers. Middle row, we have upper management of the company, and bottom row, those are the non-executive directors of the company. Collectively, these individuals, extremely talented, breadth of background knowledge and expertise, really leveraging each and every one of these individuals, plus the other team members. I mean, there's 26 odd other team members not shown here. Each one of them comes into the company with a value set that we really do appreciate, and really, really proud of the team we've assembled. So also, it's not just the directors and the upper management of the company, it's our advisors. And so our advisors are, key opinion leaders in their fields, they're luminaries. Their bios are listed on our website. Dr. Smith, he was the U.S. Department of Homeland Security Chief Veterinarian, very, very tied into the biothreat program as well. That's something where LexaGene believes we can contribute to. And Dr. Marsden, the former advisor to the White House on food safety, also the executive director of food safety at Chipotle, helped turn that company around. Dr. Rankin, she's a clinical microbiologist working at UPenn. And Mr. Stevens, who is an attorney for the food safety industry. Great, great people, very knowledgeable, big proponents of LexaGene. Quick summary here. So the MyQLab, which we've now commercialized, it meets critical needs for open access, highly multiplex testing at the point of need. We're very, very excited about the product. It does provide, of course, a rapid turnaround time, allowing people to avoid sending samples off to reference lab. It is now commercially available, and we are actively pursuing the FDA. So again, these slides are available on our website. Take a look at them at your leisure. Now I'll try to address, some of the questions here. So what are your plans for listing on the NASDAQ? Yeah, we are, the primary exchange we're on right now is the TSX up in Canada. We are very interested in the NASDAQ. Now that we're commercial, we're looking at this very, very closely. The markets are really primed, if you will, and the market can't get any hotter. And so we're looking at this very, very closely right now. No firm commitments have been made. We are having a board meeting soon to discuss this, but it is top on our mind to make a decision about the NASDAQ, and it's something that we're very looking at very, very closely. That's all I can say at this point. Let's see, how many samples can you run the MyQLab in a 24-hour period? So right now, the MyQLab, it operates, one sample at a time. And so we have designed the MyQLab so that it's small enough where people who have higher needs for processing samples can buy multiple systems. This is typical of, diagnostic companies. They are modular in nature. So if you have a need to run more samples, you can buy more systems. But right now, the system runs one at a time, and the turnaround time right now is a bit over an hour, but it's still something where people can get high enough throughput by buying multiple systems if they have that need. Importantly, again, though, the vet diagnostics market, the COVID market's not going to last forever, and veterinary diagnostics, in that particular application, they don't have a need for running 20 samples, 30 samples a day often. It's often less than that. So in that case, our form factor is perfectly suited for the vet space, and it's basically the most versatile form factor you can have across multiple markets. What are your expectations for FDA approval? And so we've just started the FDA studies right now. They are originally, later last year, we were considering going after the modern high complexity categorization from the FDA. And the reason for that is because the bar is lower. However, the FDA has been explicit in saying that they are most interested in point-of-care testing submissions. Our technology is best suited for point-of-care, and so although there are more studies for point-of-care, we've decided to go for the higher bar because those submissions are being prioritized, and it's really where LexaGene technology is designed to be operated. And so those studies will take more time. We're going to get through them as quick as we can. And we are making steady progress towards that goal. So let's see here. I got to get down to some more questions. Already answered the NASDAQ question. So in regards to detecting COVID-19 mutations and other pathogens, again, this is something where the MyQLab is ideally suited. I would argue we are the best system out there to rapidly detect new variants. Granted, for human clinical diagnostics, we have to first get through the FDA. We are working on that. We're trying to do that in as quick a manner as possible. But once we get through the FDA, we do believe that our system is going to be recognized for its value of quickly being able to detect new pathogens, which is a true value prop for a lot of clinicians. The key management of our firm in the background, I could spend half an hour talking about that. So their bios are listed on our webpage. I encourage you to go look at our website and look at each of their backgrounds. How fast are you expecting to ramp up if demand is more than manufacturing capacity? And so, a company like ours, we have to grow in a smart fashion. And so that's exactly what we're trying to do. Management meets regularly, estimating our demand for the technology, whether manufacturing can keep pace, and when to move manufacturing to contract manufacturers. We've already moved our consumable manufacturing to LaunchWorks, which is a contract manufacturer, because we don't have the capacity to meet the demand that we're anticipating early 2021. And so as the demand ramps up for our technology, we move the instrument manufacturing offsite as well. That has not yet happened, but we are making plans for doing that. We've interviewed multiple contract manufacturers, and we're preparing for that because we do expect the demand to be there. Which tests at the moment cannot be performed on this device? Create reactions inside flow cells. We position those reactions inside of a thermocycler, which has optical detection capabilities, and we perform the reaction. People have asked, are there different types of genetic tests that you can do? The answer is yes. If you have a different type of genetic test, as long as we believe we can perform it. There really are no limitations in regards to genetic tests that we cannot perform in this device. We've chosen to focus on real-time PCR because it is considered the gold standard. And I am now basically at the top of the hour. And so at this point, I'm going to respectfully, stop answering questions. If you have other questions that you want to ask myself or any other member of the team, you can contact us at info@LexaGene.com. If your questions are regarding investor relations, you can email ir@LexaGene.com, and we do try to be responsive to questions. Thank you for your support, and at this point, I'll hand it back over to the moderators.
Thank you. That will conclude the webcast. We thank you for your participation.